Hello, today we are going to study practice 4, inflammation. Here is the purpose and requirement of this lesson. Today, we will observe 3 tissue sections and several gross specimens. The most predominant change of inflammation is the exudation of leukocytes, so the most important features in inflammatory lesions is the infiltration of various inflammatory cells. First of all, let's review the various inflammatory cells. These 3 cells are neutrophils, these cells are eosinophils, and this one is basophil. The 3 cells are plasma cells, and these cells are lymphocytes, and the 3 cells are foam cells. The large one is Langerhans giant cell. And note the 4 cells. The 4 cells in their cytoplasm narrow some debris or red blood cells. These cells are typhoid cells, derived from macrophages. This is a microscopic view of peripheral blood during acute inflammation. These cells are neutrophils. They are larger than red blood cells, and the neutrophils are round. The nucleus is lobulated or rod-shaped. The cytoplasm contains small pink granules. As we have learned, neutrophils are commonly seen in the early stages of acute inflammation and in suppurative inflammation, such as acute phlegmonous appendicitis and abscess of liver. Inflammatory reactions are not neatly categorized by cell type. A variety of inflammatory cell types may be present, though one may predominate. Seen here are mainly neutrophils. But there are also plasma cells, lymphocytes, and macrophages. Macrophages can phagocytose other cells as well as cellular debris. One macrophage has pegged out by consuming a neutrophil, a red blood cell, and a nuclear fragment. Today, we will talk about 3 tissue slices: acute phlegmonous appendicitis, and abscess of liver, and tuberculosis granuloma. Okay, now the first section, acute phlegmonous appendicitis. It's a diffuse suppurative inflammation, so the features include these. At low magnification, the wall of appendix is hyperpremic and markedly swelling. The intermuscular spaces become widened because of edema. The mucosa shows erosion and extensive neutrophil infiltration. This image shows a cross-section of a normal appendix. This is the wall of the appendix. The wall is composed of 4 layers: mucosa and submucosa, muscular layer, and serosa. This is the lumen of the appendix, and here is the periappendiceal fat tissue. This image shows a cross-section of acute phlegmonous appendicitis. At low power, we can find that part of mucosa is necrotic, and the lumen is filled with inflammatory exudate. This picture shows the wall of appendix. The wall of appendix is composed of mucosa, submucosa, muscular layer, and serosa. All the layers are hyperemic and markedly swelling. The mucosa here shows erosion. At high magnification, numerous neutrophils infiltrate in the mucosa. For example, these cells are neutrophils. And this structure are glands in the mucosa. And this layer of cells are epithelial cells of the mucosa. In the submucosa, we can also find many inflammatory cells. For example, these cells are neutrophils. This is the muscular layer. The intermuscular space become widened due to edema. At high power, we can see these cells are smooth muscle cells, and the intermuscular spaces become widened because of edema. And in the intermuscular spaces, we can find many neutrophils. And note here, it's a blood vessel. In the lumen of the vessel, we can find some red blood cells and many neutrophils. These neutrophils are margination, are close to the endothelial cells. At low magnification, we can see the appendix lumen is filled with pink-stained pus. At high magnification, we can see the pus is composed of living or dendritic or necrotic neutrophils and cellular debris. The basal appendix is hyperemic and infiltration of neutrophils, and we can also find many dilated blood vessels. Now, let's make a summary for this section. The appendix lumen is filled with pus, consisting of living or dendritic or necrotic neutrophils with debris. The wall of the appendix is thickened due to a large number of neutrophils infiltration and is markedly congested. It matters. Neutrophils infiltration is throughout the wall of appendix, especially in the muscular layer. So, this is a diffuse suppurative inflammation. The features include tissue necrosis, pus formation, and diffuse neutrophil infiltration. The second section, abscess of liver. At low power, some scattered, well-defined round foci in variable size can be seen in the liver tissue. Those are abscesses. Look at the picture. This is an abscess. It's a well-defined round foci. Normal liver tissue is seen at the lower right. At the high power, in abscesses, there are a large amounts of neutrophils. Some of them undergo degenerated and necrosis. The abscess is walled off by proliferated fibrous tissue. The third tissue section is infectiogranuloma. This is a low-power microscopic view of tuberculosis. We can see several granulomas with clear border scattered in the lung. At low magnification, we can observe that the granuloma contains a central caseous necrosis and is surrounded by mixtures of cells including epithelioid cells, non-Hansch's cells, and lymphocytes. The caseous necrosis material appears as amorphous, structureless, and eosinophilic granular debris with complete loss of cellular details. Around caseous necrosis are some epithelioid cells and scattered non-Hansch's cells. These cells are epithelioid cells. They are derived from macrophages and quite resemble epithelial cells, so we call them epithelioid cells. The boundary of these cells are not clear, and the nucleus is round or oval, and staining is very light. The cytoplasm is light and rich. This cell is a non-Hansch's cells. Non-Hansch's cells is derived from the fusion of epithelioid cells. The cell has large cell body and multiple nuclei. The nuclei are arranged in garland or horse hoof form or assembled together, and quite resemble those of epithelioid cells. The cytoplasm is red stained and abundant. Look at the picture. This is a well-defined granuloma. It's composed of a central caseous necrosis and epithelioid cells and some lymphocytes. And in this area, we also can find some alveoli, but these alveoli are not normal. They are filled with edema fluid and some inflammatory cells. Here is caseous necrosis. It's pink stained and amorphous. These cells are epithelioid cells, and at the rim of the granuloma, there are some fibroblasts. The two cells are non-Hansch's cells. When the lesion increases or expands, the lesions can fuse to form confluent tubercles. Okay, now let's move on to the second part, gross specimen observation. This is an acute severe hepatitis. The liver decreased in size. It's significantly shrunken and deformed. On the cut surface, the liver shows yellowish green or dark red appearance. Necrotic areas have a muddy red, mushy appearance. This specimen is fibrinous pericarditis. The pericarditis surface is covered by a layer of gray-white fibrinous exudate, which give a shaggy appearance. This is another fibrinous pericarditis. There is a knot of fibrinous exudate on the epicardial surface. The hearing heart is formed by the rubbing of the fibrinous exudate during the movement of cardiac pulsation. The mucosal surface of trachea and bronchi is covered with a thick and gray-white membrane. That's situ membrane, part of which have been detached, even causing blocking. This gross specimen is cerebral abscess. On cut surface of the brain, it shows an abscess with a clear edge. The abscess enveloped with a layer of thick fibrous capsule. The cavity of the abscess is filled with the yellowish or gray-red pus. The specimen is multi-abscess of liver. On the cut surface of the liver, we can find several well-real-sized and irregular cysts resulting from liquefactive necrosis of the liver. Some cysts contain solid of fluffy gray-white pus after fixation. Some cysts are empty because the pus is lost. This is a normal appendix, and the three appendix are not normal. They are acute phlegmonous appendicitis. The three appendix are markedly swollen, and the serosal surface is hyperemic and partially covered by gray to yellow suppurative exudate. And let's note here, we can find mainly dilated blood vessels. This specimen is a normal brain, and this one is acute pyogenic meningitis. The meningeal vessels are highly dilated and hyperemic. A thick layer of purulent exudates covers on the meningeal surface. The gray-yellow purulent exudates fill subarachnoid space and cover the sulci and gyri, so that their structures are unclear. This one is a normal tonsil, and this gross specimen is chronic tonsillitis. The tonsil is enlarged obviously. It's firm and gray-white. The capsule isn't smooth, and exudate is not obvious. This gross specimen is chronic cholecystitis. Chronic cholecystitis is a general proliferative inflammation. The gallbladder is enlarged, and the wall was significantly thickened due to fibrosis. There are 4 irregular gallstones in the gallbladder.